Excellent. So our next speaker is Elizabeth Burgener. She's an instructor in pediatrics and pulmonary medicine, has MCHRI support with a clinical trainee award, and she will be telling us about filamentous bacteriophages and cystic fibrosis.
Thank you for the introduction, and thank you for allowing me to present my work today. And again, thank you for my trainee grant during fellowship that supported this work. So today I'm going to be talking about PF bacteriophage and clinical outcomes in CF. So cystic fibrosis is an autosomal recessive fatal disease. It's caused by a mutation in the Cftr protein, which is a chloride channel. It's present throughout the body, so it is a multisystemic disease. However, the majority of morbidity and mortality comes from pulmonary disease. That's what I'll be focusing on today. When patients with CF are born, their lungs are structurally normal. However, they quickly develop really thickened mucus, which then becomes infected, immune cells influx, and then you end up with a vicious cycle of infection and inflammation that we really can't break. And Pseudomonas is a gram-negative bacteria, which is a bad player in CF. It's the most commonly cultured organism from CF sputum. We see it more increasing with age, and it's very difficult to get rid of, partly because it forms biofilms and also because it's very resistant to antibiotics. And we know that once patients have Pseudomonas, they have a faster decline in lung function and a higher mortality. So PF bacteriophage, and PF stands for Pseudomonas filamentous phage. And so it's a bacteriophage that's present in Pseudomonas. This is not a talk about phage therapy. This is not something we're giving to patients. This is something that's present in Pseudomonas. And the story I'm going to tell you today is about how we think it's actually more of a virulence factor of Pseudomonas and actually helps set up infection and promotes chronic infection in Pseudomonas. And so it's a filamentous phage. So it's long and skinny. It's basically a single strand of DNA surrounded in a protein coat. It infects Pseudomonas. It incorporates its genome into the bacterial chromosome, so it's a prophage. And then Pseudomonas produces many copies of it and then extrudes the phage, but it does not kill Pseudomonas. So the Pseudomonas continues on and makes more phage. So this is considered lysogenic replication, not lytic. And we know that when phage is present in a biofilm, it actually makes it liquid crystal. Or liquid crystal structure. So in panel A there you can see two colonies of Pseudomonas. The top does not produce phage, where the bottom one does, and you can see that that bottom one can polarize light, so it's actually birefringent. And in panel B, this is looking at CF patients' sputum. Those with Pseudomonas have higher birefringence or more ability to polarize light than those without Pseudomonas. And you can, if you add phage to the Pseudomonas negative sputum, you can actually match the birefringence. So we have Pseudomonas producing this phage. It interacts with the extracellular matrix, or in this case, sputum. And from what we know from work that's been done in the lab, we know that it's more birefringent, it's more viscous, it's more adherent, it sequesters antibiotics, and it inhibits phagocytosis. So all of these things we hypothesized would promote chronic infection or make patients sicker. This is a video of adding a drop of phage to hyaluronic acid, which is a very negative polymer, and you can see that it causes the hyaluronic acid to organize into this higher order structure that's very liquid. And so this is happening in our patient's sputum, in their lungs. So when I came on board as a fellow about 3 or 4 years ago on this project, we wanted to apply this to CF, and our hypothesis was that patients with CF, with Pseudomonas with this phage, would be sicker or have worse outcomes than those patients that have Pseudomonas but without this phage. So we first started broad and looking just to see if this phage is out there. So we looked at the Pseudomonas Genome Database, which is an online database where investigators have basically logged their whole genome sequencing of Pseudomonas. And as I mentioned, it's a prophage, so it incorporates into the Pseudomonas chromosome. So if you have whole genome sequencing of Pseudomonas, you can look for this phage. So in this database is not just CF, it is clinical isolates, it's environmental isolates, it's many different things. So we looked for specifically the gene for the coat protein of the phage, as that's necessary for phage production. And we saw that in this database of about 2,200 Pseudomonas genomes, 52% contained this filamentous phage. Then we looked closer at a well-characterized Danish cohort of CF patients, and this was 34 pediatric patients with known chronic Pseudomonas followed over a period of about 10 to 12 years with multiple isolates from each patient. And in those 474 isolates, 44% contained our PF phage. And then I collected sputum from a cohort of Stanford patients and basically took sputum from patients that could expectorate. And then on the sputum performed qPCR looking for the specific phage. So a different method in looking for the phage than the other two cohorts. But we found in 58 patients that had Pseudomonas in their sputum that 36% of our patients here at Stanford had PF phage in their sputum. Panels B and C are looking closer at the Danish cohort, and because the Danish data was longitudinal, we categorized patients into phage negative, phage positive, or intermittent. And you can see that our phage positive patients are significantly older than our phage negative patients. Panel C is also looking at age, but in a different way. This is looking at attained age, so the age at the end of the study, and then the proportion of their isolates that contain phage. And you can see the older the patient, the more likely they had more isolates with phage. In looking closer at our Stanford cohort, we saw that the levels of phage and Pseudomonas were tightly correlated, and that that phage was associated with chronic infection. So in panel A, this is the log of phage copies versus the log of Pseudomonas copies. You can see they're tightly correlated with an R of 0.84. However, that line is shifted up. It's not on that the dotted line. It would be a 1-to-1 relationship. So we know there's more phage than there is Pseudomonas, on the order of 1 to 2 logs. And then panel B is when we applied the Leeds criteria, which is a definition for chronic Pseudomonas infection in CF, and you can see 100% of our patients with phage met criteria for chronic infection. And next we looked at antibiotic resistance. So our patients that carried PF phage had significantly higher incidence of resistance to amikacin, S-treonam, and meropenem. And we knew from some of the in vitro work that phage could be sequestered by, that antibiotics could be sequestered by this liquid crystal. So we suspected that this was what was going on behind it. So again, we did another sequestration assay with these specific antibiotics, basically took a dialysis cassette with both DNA and phage and concentrations to produce a liquid crystal, added the antibiotics of choice, you catch what comes through, and then expose sensitive E. coli to the dialysate, and the amount of E. coli that survives is a measure of the amount of antibiotic that was sequestered. And our 3 antibiotics where we saw clinical significance, amikacin, meropenem, S-treonam, were all sequestered, with ciprofloxacin, which we didn't see clinical significance, was not sequestered. So putting it all together, we think that phage within the biofilm is sequestering antibiotics, protecting Pseudomonas from the antibiotics, essentially exposing it to subtherapeutic levels and promoting the survival of resistant clones. I've continued to follow these patients over time, so everything that I presented previously was cross-sectional at the time that they entered our study, but I've been following these patients for about 2 years now, and so we have 24 phage-positive patients with multiple encounters and 25 phage-negative patients. And when we look at lung function over time, we see that our phage-positive patients have a significant decrease in lung function over time, while our PF-negative patients don't. And I mentioned earlier, we know Pseudomonas is associated with the decline in lung function, but I think this data may argue that phage is maybe could be responsible for that or more responsible for that than the Pseudomonas itself. When we look specifically at lung function versus the burden of phage or the level of phage, we see that the higher the phage copy, the lower the lung function. And this is using a model of mixed linear regression where we're adjusting for ages at sampling. So in conclusion, we know that PF phage in patients with CF is abundant in the sputum of a subset of our patients with Pseudomonas aeruginosa. It's associated with chronic Pseudomonas infection within the airway. It's associated with antibiotic resistance and associated with the decline in lung function over time. And we think that phage may represent a novel therapeutic target so that we can treat or even prevent chronic Pseudomonas infection. And with that, I'd like to thank my 2 main mentors, Dr. Paul Boyke and Dr. Carlos Mila, and their labs and teams that have been very helpful in completing this research, as well as my funding sources. Thank you.
Are there any questions?
What is the cause of the Phage increase? Is it just the age of the individual, the chronology of their condition, or does it relate to many antibiotics that may change the dynamics of the Pseudomonas to become more Phage-like?
Yeah, I can't give you a direct answer because we don't know yet, but I think that the strains that produce phage are more likely to set up chronic infection, so I think we're more likely to see it in our older patients. What I can tell you is from that Danish cohort, we looked at strains within a patient over time. The patients that were intermittent, where phage seemed to come and go, it wasn't that they had the same strain of Pseudomonas and the phage was coming and going, it was that we were seeing different strains. So I think it's more strain specific, but I think that it's more likely to set up chronic infection if phage is there. And I think our older patients have had more exposure and are more likely to have chronic infection. But that's my speculation.
And has it changed with the more novel transporter treatments that provide better lung maintenance?
Yeah, with our Cftr modulators. It's too early to say. I mean, the Cftr modulators in the first one came out in 2012. I didn't start studying this until 2015, 16, so I don't have any data from before. But what we know from the data on the early modulators is that we see a decrease in Pseudomonas initially when patients take them, but then eventually the Pseudomonas counts come right back up. So it doesn't seem like it's playing a huge role in infection, but what it's playing with phage, I can't speculate yet.